Hello everyone, hope you all are doing well. So today we are going to discuss topic 28 of paper 2 topic 28. So here we are going to discuss breeding for disease and pest resistance. Breeding for disease and pest resistance. So this is the topic 28 of paper 2. So the first topic of this session is mechanism involved in insect pest resistance. The first topic for this session we are going to discuss is mechanism involved in insect pest resistance. So the first one is non-preference. So as the name suggests, हम अपने आप को ऐसा बना लेंगे कि कोई हमें प्रीफर ना करे. ओके हम अपने आप को अनअट्रैक्टिव, वी विल मेक आवर सेल्स अनअट्रैक्टिव, अनडिजायरेबल, सो दैट नो वन प्रीफर्स अस. So here insect will not prefer us. So it makes the host undesirable and unattractive to insect for food, shelter and reproduction. For food, shelter and reproduction. So when the host will become undesirable and unattractive, the insect will not prefer to that host plant. It is also called non-acceptance or antigenosis. You should remember this term. So here what happens? Color of plant, light penetration, hairiness, leaf angle, odor, taste, etc. So these are the characteristics of the plant that become unattractive. So these characteristics are associated with non-preference. As these characteristics will become undesirable and unattractive, the insect will not prefer that plant. So for example, you can see in wheat, for the insect bollworms, the wheat's leaves become smooth leaves, long pedicle, thick ball reed. So these characters are not preferred by bollworms, so they do not attack on wheat. For you can also see it in pea, for pea aphid, for pea aphid. So here we see that the leaves of pea turn to yellow green leaves, which is not preferred by aphid. So this was all about non-preference. Hope you all understand this. Now moving on to second one, that is antibiosis. This was antigenosis. Now second one is antibiosis. Number 2, antibiosis. It refers to the adverse effect of host plant on development. Adverse effect of host plant. Effect of host plant on development of reproduction of insect pest, which feed on resistant plant. So in simple word, we can see that it has antibiosis means showing adverse. The plant shows adverse effect on development of pest. So here, resistant plant retard growth rate of reproduction of insect pest. Here the plant becomes resistant, and the resistant plant retard growth rate. It slow down the growth rate of reproduction of pest, and hence it is antibiosis is also called true form of resistance. True form of resistance. For example, you can see in cotton that in cotton antibiosis is related with high level of gossipol. Cotton antibiosis is related with high level of gossipol, high level of tannins, heliosides and silica content. The high level of these materials helps the plant to resist the insect pest and retard its reproduction or growth rate. So this was all about the antibiosis. Now moving on, the third one is tolerance. Third mechanism is tolerance. As the word says, to tolerate. So here the ability of plant or variety to produce greater than susceptible variety. Here it is the ability of plant or variety to produce greater than susceptible variety. The tolerant variety will always produce greater than susceptible variety at same level of insect attack, equal to same level of insect attack. So the tolerant variety will produce more than susceptible variety at same level of insect attack. And these varieties also have greater recovery of damaged parts than susceptible varieties. Greater recovery of damaged parts. The damaged parts recover faster as compared to susceptible variety. Example, you can see hybrid cotton. So the levels of production doesn't decrease after insect attack also in hybrid cotton, hence it is a tolerant variety. Hope you all understand this. Now moving on to the last mechanism, that is escape. Last mechanism number 4 is escape. It refers to avoidance of variety from insect attack. Avoidance. The variety avoids from insect attack. Avoidance from insect attack due to earliness on its cultivation in the season where insect population is very low. Due to earliness. So it reproduces on that season where insect population is low, and when the insect population increases, at that time it has escaped because it has already produced, or it has its cultivation or its reproduction or vegetative phase has gone, earliness. So it avoids through earliness. For example, you have heard about early maturity cotton crop. They escape pink bollworm infestation, which occurs late in season. They escape pink bollworm infestation, which occurs in late season. So due to its earliness, it escapes pink bollworm infestation. Hope you all understand this. Now moving on to next topic of this session, that is gene pyramiding and gene interrogation for disease resistance. Gene pyramiding and gene interrogation for disease resistance. So we will discuss here gene pyramiding and gene interrogation. First of all, we are going to discuss gene pyramiding. So it refers to the incorporation of two or more genes. It refers to incorporation of two or more genes or two or more major genes in host for specific resistance to a pathogen. Under it, all major genes are incorporated in a single cultivar. All major genes are incorporated in a single cultivar. So it has various merits and demerits. First we will discuss merits. It is preferable over multi-lines. Preferable over multi-lines, where each major gene is incorporated into separate genotype and then resistant genotype are mixed together. Here what happens? Incorporated into separate genotypes. And then resistant genotypes are mixed. But in gene pyramiding, in a single genotype, all are incorporated. Here, the combination of several major genes for a specific resistance provides protection against several new races, as there are combination of several major genes. Combination of several major genes leads to protection against several new races. Protection against many new races. New variety have durable resistance. As new variety is produced, so there is durable resistance. It has been used out in against crown rust. It has been nicely used in crown rust. So these were merits. Now moving to demerits of gene pyramiding. Demerits. Needs a lot of effort to incorporate several genes. High effort is required to incorporate several genes. It requires extensive testing. This approach will enhance chances of formation of new virulent races of pathogen, can lead to formation of new virulent races of pathogen. The agronomic performance of new cultivar is restricted to the recurrent parent when backcross is used. So here agronomic performance is restricted to recurrent parent when backcross is used to incorporate genes. So these are the demerits of gene pyramiding. Now we will move into gene introgression. So we have finished gene pyramiding. Now what is gene introgression? Gene introgression is the transfer of genetic material from one species into gene pool of transfer of genetic material from one species into gene pool of another by repeated backcrossing of interspecific hybrid with one of its parent species. It is a long-term process. Even when artificial, it is a long-term process. Although it is artificial, but it is a long-term process. It may take many hybrid generations before significant backcrossing. It may take many hybrid generations before significant backcross occurs. It results in complex, highly variable nature of genes. And may only involve a minimum percent of donor genome. Significant and observed in several plants such as tomato. So this was about gene pyramiding and gene introgression. Hope you all understand this. Now we will move into the next topic of this session, that is gene for gene hypothesis. Gene for gene hypothesis. It was given by Floor in 1956. On his studies of host interaction in flax or linseed. For rust caused by Malump sorreline. It is also known as Floor hypothesis. It states that the gene for gene hypothesis states that for each gene controlling resistance in host gen, for each gene controlling resistance in host, there is a corresponding gene controlling pathogenicity in pathogen. There is a corresponding gene containing pathogenicity in pathogen. So for each gene controlling resistance in host, there is a corresponding gene that contains pathogenicity in pathogen. The resistance of host is governed by dominant gene. This is governed by dominant gene. Whereas the pathogenicity in pathogen, virulence of pathogen, is governed by recessive gene. The genotype of host and pathogen determine disease reaction, and the genotype of both determine disease reaction. When genes in host and pathogen match for all loci, when these genes, the genes in host and pathogen match for match for all loci, when these genes match for all loci, then only host will show susceptible reaction. Then when only host will show susceptible reaction. If some gene loci remain unmatched, the host will show resistant reaction. When the when the genes in the same loci remains unmatched, the host will show resistant reaction. So I hope you all understand this. What happens here? For genes in resistance, genes of resistance in host, there are corresponding genes of pathogenicity in pathogen. When these two genes match for each other in loci, then only susceptibility is shown. When these two genes do not match, then host will show resistance. I hope you all understand this. Now, what are the models of floor hypothesis? Models of floor hypothesis. First one is single gene for gene hypothesis. Single gene for gene hypothesis. Single gene. Here there is single gene from host and pathogen. Single gene from host and pathogen. Secondly, double gene for gene hypothesis. Double gene for gene hypothesis. Here, two pairs of gene are involved in host pathogen interaction. Two pairs of gene involved in host pathogen reaction. So when both the pair match, then only the reaction will be susceptible. When both pair not match or one of the pair doesn't match, then the reaction will be resistance. Now third one is multiple gene for gene reaction. Here three pairs are involved, three pairs, and if all the pair match, then the reaction will be susceptible, and if all the pair or any one of the or all the pair do not match or any one of the pair do not match, then the reaction will be resistance. Hope you all understand this. So this was all about gene for gene hypothesis. So that is all for today's. For any queries, do comment. If you like it, please press the like button, do share and subscribe, and you can also join us on a Telegram for better discussion or any discussions related to agriculture, optional or UPSC. So the Telegram channel, you can go through the link which has been provided by about section of this YouTube channel. So you can go there and follow the channel on the Telegram also for healthy discussions or resolve of queries. So that's all for today. Have a nice day. Thank you.